Hi, all.
Hello.
How are you
Hello.
all? Doing
Good.
good?
Tired.
Tired?
Yep.
Me too.
Me too.
I just came back from department. I had lab, Group B lab.
I've been
So,
getting or having to set up for a job while getting all this stuff done. I mean, it's been great, but whenever you start working for a hospital, they make you go through the ropes.
yeah. Ah. Okay, so let me see what's the time. Okay, so just wait another 1 minute, then we will start. So you started working on Labstar?
No, I'm actually going to do that tonight after I'm done with this session.
Me too.
I started working on it. My computer glitched up so bad.
Oh,
Yeah.
what happened?
I had to go back. It lets you reload. So you go through like a module and then it lets you reload through it. So I'm fine. It saves where I'm at, but it's just taken me a while to get through it.
Hmm.
Can you retake the Labsters as many times as you need to?
Labstar? Yes, you can. Yeah. But every time you have to spend that much time. If you finish within 1 hour, so you have to give extra hour. So better, before even you start, know the concept, read the theory, okay? Then start working on the simulation. So it will save time. Okay, I think we need to start. So I started chapter 3, right?
Yes, ma'am. I believe you're on slide 9, Koch's postulates.
Oh, okay. Okay. So I talked about, oh yeah, I talked into symbiosis theory. And I talked about this slide also, right? So I need to start from here. Can you see my slide?
Yes,
Yeah.
ma'am.
Okay. Okay. So let me fix my pen. Okay. So Koch postulate, Koch actually gave us few rules, okay? 5 postulates are there actually. He talked about, how to identify the microbes, okay? If a person is carrying the microbes, or on the other way, you can say like this. So if a microbe is responsible for a specific disease, then that microbes must be present inside the body of that person, okay? Not only that, uh, you can reculture the microbes, and you can reintroduce that microbes into somebody else's body, and you can infect somebody else with the same microbes with the same signs and symptoms, okay? And that's how you can actually identify, and you can know about the microbes, like which microbes create what type of disease and what are the signs and symptoms. So to identify all those, a Koch actually gave us 4 postulates, okay? We still follow all these 4 postulates to identify the disease, okay? So the first postulate is the microorganism must be found in abundance in all the organisms suffering from the disease, but should not be found in healthy organisms, okay? So like if you have influenza or like corona, so that means COVID, actually not corona. So that coronavirus or influenza virus must be inside the body, okay? And not only that, if 100 patients, 100 persons, or it could be million of persons who are infected with COVID disease, COVID-19 disease, or maybe the influenza disease, if we examine their blood and in every cases, actually, we can find out that coronavirus or that flu virus inside all those persons. So basically what he meant that one specific disease is actually caused by a specific microbes. Okay, so he told that one disease means equals one microbe, and in all the cases the signs and symptoms should be same. In each and every cases, the signs and symptoms are same. Okay, so that's one. Okay, this is it. So that's the first postulate. So he told that if we take that person, then from the blood we actually can collect the specimen. So all the person who are having the same signs and symptoms, if we collect any body fluid, it could be blood or anything, any body fluid, then each and every cases we are actually going to find out that same microbe. Okay, so that's the postulate one. Second, what he told, the microorganism must be isolated from a diseased organism and grown in pure culture. And in each and every case, if we collect the blood from all those sample person, then that microorganisms will be there. And he gave one condition: we can find out those microbes or that microbes only in the diseased person. Healthy person should not carry. Okay, at this moment, in present time, we have so many objections, and we can talk about it later in, I think, chapter 4 or chapter 5 about this. But that's the second postulate. So basically, with same signs and symptoms, or you can say with the same disease, it could be only one microbe who is responsible for all the cases. And if we collect the sample, body fluid sample from that person, for each and every one, that same microbe should be there. Now, not only that, the second one, we can actually, from that blood, if we collect that blood, we can actually culture the specimen. We can regrow the specimen. So basically what you can do, you can take the body fluid, any type of body fluid, maybe blood or urine or whatever, in a slide or in a petri dish. Okay, so this could be the blood, this could be the blood, and you can actually culture that microbes inside the lab. Okay, so basically you can regrow. Now, so this is the second batch. Okay, so first batch of microbes you collected from an infected person's body. So this is your second batch of specimen. Now, if you inject this second batch of specimen into a healthy person, okay, at this moment we are just injecting all these microbes into one healthy person's body. If you inject that, this person will be infected with the same disease. Not only that, that same signs and symptoms will be here for that healthy person. Okay, so that actually proves that this specimen and these specimens are the same microbes. So basically, in each cases, or all the cases, for all the cases, This is the microbe who is responsible for one particular specific disease with specific signs and symptoms. Okay. Now, not only that, the fourth one tells that when we collect the sample, but again the blood sample or urine sample from that person. So this is our test material. This person is our test material, right? So if we collect the blood sample from this test material, and we take it inside a slide or petri dish and we want to culture, okay, we can find out the same specimen that we got over here. So these are the four postulates. These are the steps by which we can diagnose the disease. We still do that. When you are sick, when you go to the doctor, doctor does that. If it's your, okay, so tummy pain, doctor collect your urine, try to culture the bacteria and try to find out the disease. So these are 4 rules or 4 steps to identify the specific bacteria or specific microbes, you can tell. So these are the Koch postulate. We still follow all these steps. The main thing, what actually Koch wanted to tell is one microbe is responsible for one disease. So this is the main theme of this Koch postulate. Okay, every time one microbe is responsible for one specific disease, and each and every cases, the signs and symptoms will be the same. So this is the historical foundation. Okay, so I forgot to take this out. Let me move this one, otherwise. Okay. So this is the historical foundations. I already talked about, I think about all of the scientists. So Robert Hooke first discovered the cell. Not only that, he made one very simple microscope. Simple microscope means that had only one single lens. Okay. Then Leeuwenhoek made that also simple microscope, but the powerful microscope, and we first observed the living cells. Okay. Then Pasteur actually he supported that because of the microbes, okay, all the diseases are going to be have occurred, or on the other way, that microbes are very small organisms who are responsible for the diseases. He found out that why the fermentation of his wine and all those. So that's the Pasteur. Semmelweis, he was the surgeon. I talked about it last week, that he noticed that all the women immediately after giving birth, they used to get that puerperal infections because of the medical students. They used to do autopsy, and all those immediately after doing autopsy, they used to check all those mother, and that was the cause of the lots of infections. Lister, Joseph Lister, he actually started using carbolic acid. He was a surgeon, and he noticed that the percentage of patient who is to die immediately after surgery was very high, but he started using that carbolic acid, okay, a phenolic compound. After started using that, he noticed that that percentage was slowly going down. and then after that onwards, he started using, you know, disinfecting all the surgical equipment, not only that surgical area. Maybe the surgery would be on the tummy, so he used to disinfect tummy. And coach, he told that one microbes is responsible for one disease. So because of all these scientists' contribution, that actually made, or you can say that actually cut the path of our modern understanding about the microbes. So cell, bacterial cell, or you can say the over here it's prokaryotic. Prokaryotes mean, when we are talking about prokaryotic cell, means we are actually talking about both bacteria and archaea. Okay, so these are few common shapes. The coccus, round. Then bacillus, so coccus, round. Bacillus, that's the rod-shaped, and we have spidilla or spirillum. So these are the three most common shape of the bacteria. Now, bacteria, mostly the prokaryotes, they have different arrangement. Okay, sometimes two round cells, they remain together. Under the slide, if you observe, you can see that every time the two cells are together, if this is the arrangement, then it is called diplococcus. When four cells are together, so over here we are talking about just one plane. This is also one plane, but when it is tetrad, when there are four cells, means the cells are arranged in two planes. Okay, so that's called tetrad. Streptococcus, the word strepto, okay, any word that starts with strepto, that means it's chain. Okay, coccus means round and strepto means chain. So basically all the round shaped bacteria, they are in chain. Staphylo means cluster. Staphylococcus, every time Staphylococcus or arrangement, you can see that in lab, if you see Staphylococcus epidermidis, they make a large grape-like cluster, and it look like a really one bunch of grape. Okay, so over here you can understand easily that's the multiple plane. Okay, the division of cells occurs in multiple plane. So that's the Staphylococcus. So these are the very common arrangement of prokaryotes, both for bacteria and archaea. In lab, we are actually going to work with the bacteria, so you can see all these arrangement. Same way, when it is a streptobacillus, means it's a long chain of rod-shaped shell. There are few cells, few bacteria. Those cells, they are actually rod-shaped, okay? But they have a kink or bend at the center. So they looks like curved rod. So they are called vibrio. Coccobacillus, they are actually rod-shaped bacteria, but they are the rod is so small, they looks like coccus, but they actually are not coccus, they are actually rod-shaped bacteria. In that case, that shape is called Coccobacillus. Spirillum, spiral-shaped. So these are the arrangement and the different shapes of bacteria. So basically three different shapes, coccus, bacillus, and the spirillum. And these are all different arrangement. Okay, diplococcus, tetrad, streptococcus, very important, staphylococcus, or the cluster of the cells. Now the prokaryotic cell structure. So from now, we are going to work on the different organelles and cell structure of prokaryotic cells. So this is the picture of a prokaryotic cell, I think, yeah. So if we go from outside to inside of a prokaryotic cell, so these are the cell organelles or the parts of the cell we must see. Okay, so the first is the appendages, and then we are going to observe the glycocalyx. So okay, so from outside, first organelles are the appendages. So we can see appendages over here outside of the cell. So one long flagella, this one. Flagella, one long flagella. Then small hair-like structure surrounding the whole outer surface of the prokaryotes. Those are the fimbriae, very thin and very small, little larger than fimbriae, but smaller than the flagellum, a little longer structure, hair-like structure, that's the pilus. So three different types of appendages are there. This is the flagellum, the longest one. This is the locomotory organ. It helps to move bacteria. Also, at the same time, it helps bacteria to attach. So basically, it's locomotory and attachment organ. Fimbriae, mostly the attachment organ. Okay. Pili or pili or pilus, they are actually sex organ and the attachment organ. Bacteria, sometimes they have sexual reproduction. It looks like sexual reproduction because two bacteria get involved into the process. Okay, so that's why it's almost like sexual reproduction, but it's not sexual reproduction truly. But during that process, it's called conjugation, and that time this spiders, they actually exchange their chromosome materials through the pilus. Okay, so this is their sex organ. So these are the appendages. Then immediately beneath the appendages, we can find capsule. Okay? Capsule, basically two different types. Either it's called glycocalyx. Two different types. The first one is, you can say, capsule, and little slimy actually. That's called slime layer. So glycocalyx, let me write it down. Glycocalyx, that's actually jelly-like outer coverings of prokaryotic cells. Archaea, they don't have that much glycocalyx, mostly for the bacteria. It's jelly-like substance. Okay. Now they are basically, they're the protective organ. Okay, number one. Number two, adhesive organ. They help the bacteria to remain attached with something. They determine the pathogenicity of the bacteria. Okay, like you will see the bacteria without this glycocalyx, we can kill them easily. But the bacteria with glycocalyx, it's very hard to kill them. So this glycocalyx, it actually determines the pathogenicity of the bacteria. Now this jelly-like layer, you can see this one. It's actually two different types. Okay, one type is called capsule, and the second type is called slime layer. So both are same. There is very little difference. Only thing, the capsule, it's tightly organized. Okay, very regular in arrangement. We can say regular arrangement. So if there is a capsule outside of any bacteria, so everywhere you can see that jelly-like substances. Okay, but the slime layer, they are very, very loosely arranged. So basically, if it is slime layer, what you can see if you observe the bacteria from outside, you can see. So maybe this portion, it is covered with the glycocalyx. Over here, there is no jelly layer, no slime layer, okay? So that's why I say that it's loosely arranged. The arrangement is very loose and irregular. Otherwise, there is no difference between the capsules and the slime layer. Both, that's why both type of outer coverings are called glycocalyx. When it is hard, little harder than the slime layer, and arrangement is regular, that is called capsule. When it is loose, then that's the slime layer. Now, if you go beyond that, go inside beyond that glycocalyx, immediately beneath that capsular slime layer, you can see the cell wall, okay? So this is the cell wall, that white portion, that's the cell wall. Cell wall actually help bacteria. Cell wall prevents the osmotic pressure. It helps, it protects bacteria from the osmotic pressure. It gives the shape of the bacteria. It's almost kind of protective organ. Now, beneath that cell wall, we can see the plasma membrane or the cell membrane, this brown one, this one. That's the plasma membrane. Plasma membrane controls all the chemical activities that goes on inside the bacterial cell, inside the prokaryotic cell. Everything is controlled by this plasma membrane. These are semi-permeable membrane. Like this membrane decides which particles or what are the ions can come in or who can go out. So inside, all the materials are controlled by the plasma membrane. So very, very important part. Not only that, all the metabolic activity actually goes on inside this plasma membrane, the protein of the plasma membrane. So this part, this plasma membrane is very important for the prokaryotic cell. It's not like our nucleus. For eukaryotes, for us, the most important organelles, you can say, who actually control the activity is the nucleus. But over here, for the prokaryotes, plasma membrane actually mostly control. Now, immediately beneath the plasma membrane, we have another jelly-like semi-solid, you can say, the cytoplasm. Almost fluid-like structure. All the organelles, different type of organelles, they float inside that cytosol or cytoplasm. So cytosol or cytoplasm that carry the organelles. Prokaryotic cell, they don't have any nucleus and they do not have any membrane bound organelles, okay? But we can find that there is a spot, specific area where we can see the chromosomes, okay? That area is called nucleoid. So this is the nucleoid region of prokaryotic cell where we can see the chromosome, the genetic material. Now, small dot-like structure, these are the ribosomes. Remember that the first day I told that prokaryotic cell, they do not have, two things are very important about the prokaryotic cells. They do not have nucleus, okay? And they do not have any membrane-bound organelles. So basically, there is no such organ that has membrane, that is covered by membrane, like nucleus, okay? It does not have nucleus because to be a nucleus, it must have that plasma membrane or the membrane, any type of membrane. So that's the reason it does not have membrane. It does not have, you can say, mitochondria like us. Okay, So no membrane bound organelles, but it has ribosome, small dots, that's the ribosome. Ribosome is responsible for protein synthesis, okay? So ribosome, prokaryotic cell carry ribosome. Ribosome has membrane, okay? But somehow scientists does not agree that ribosomes are the membrane bound organelles. I don't know, still the controversies are over there, but that's the only membrane bound organelles prokaryotic cell have. That's the only one, the ribosome. So this is the main structure, you know, the overview of a prokaryotic cell. You can see the inclusions bodies. There are different inclusions body. You can see cytoskeleton. Cytoskeleton, previously when I studied, when I did my postgraduate degree and all those, that time we did not have any idea about that prokaryotic cell has cytoskeleton. But recent study, they shows that prokaryotes also carry the cytoskeleton. Cytoskeleton for us, for eukaryotes, it actually transport material inside the cell. That's one structure. Not only that, they are the endoskeleton of a scale. Okay, the cells rigidity, cells shape, everything is controlled by the cytoskeleton. So nowadays we know that they have, prokaryotic cell also has cytoskeleton and almost the same function like eukaryotes. Endospore, there are few bacteria who has endospore, but not all prokaryotic cell carry endospore. Okay, so this is the now we will go into the detail of each and every structure. First, prokaryotic cell walls. So cell walls, what it does, as I told, the cell wall, you can see that we have capsule or slime layer, means glycocalyx layer, and even before glycocalyx we have different type of, you know, the flagella, pili, or fimbriae, different type of appendages. Okay, immediately beneath the the glycocalyx layer we have cell layer, cell wall. Okay, and that white one over here, this one, and just beneath the cell wall we have plasma membrane. So even before the plasma membrane we have cell wall. So what's the function of the cell wall? And we all know that most of the time all these microbes, they remain, most of the time they remain inside the water or in humid area. That actually help them to remain attached with a solid substance. Okay, that help them to provide their food, that help them to get their oxygen. So humidity is very important actually for all prokaryotic growth, prokaryotic cell growth. So that cell wall, first of all, it gives the shape, okay, shape of the cell. That's there. The other thing is the protection from osmotic pressure. So osmosis, so two things first you need to know. One is called diffusion and osmosis. So diffusion, what happened, let's say this is a room, okay, and if I spray any air freshener over here, the person sitting all the way at the end, after a certain period of time, that smell will go all the way over there. Okay, so the whole room, at certain point, the whole room will be, you know, anybody can get that smell of the air freshener. So what happens over here? So what happens, this is the tendency of matter, any matter, solid matter, you can say, they move from high concentration to low concentration. Okay, that's the tendency of any solid particles, not the matter, particles. So same thing happens over here when you spray that room freshener. So that's the concentrated area. So the concentration is high over here. That's the reason all these organic chemical particles, they actually spread evenly throughout the whole room. And that's why everybody actually from any corner of that room can get that smell. So this process, when the particles move from high concentration to low concentration, this property is called the diffusion. Okay, very, very important. You really need to understand this process. Otherwise, this slide will be very difficult to understand. So when we are talking about diffusion, we are talking about the movement of particles, any particles that moves from high concentration to low concentration. And when they move, we really do not need to spend any energy, any ATP. Okay, they can move very, without any cost, and they will move until there is a balance. So these particles will move all the way from here to here until they are evenly distributed. Okay, the concentration are same on both the area. So that's the diffusion. When it is a solid particle, when it is a movement of solid particle, we call that's the diffusion. When it is a movement of liquid or movement of water, then that is called osmosis. Okay, so same thing, osmosis is same thing. So in this case, what happened? Water actually moves from high to low and without utilizing any energy. So these are the two very important definitions you need to understand. Now the next thing, if I ask you to make a solution, I can tell you make 100% sugar solution. Okay. So to make that 100% sugar solution, what you can do, either you can add 70% sugar and 30% water. Okay. Or you can do 30% sugar plus 70% water. Or you can do 40% water, 60% sugar. So in all the cases, you are actually going to make 100% sugar solution. So two things you added. One is the solid material. Okay, that's the solute, that is the sugar. And another one is the solvent into which you are going to actually dissolve the solute. So that's the solvent. Over here, water is the solvent and sugar is the solute. So that's how it goes. So from this picture, you can easily understand that if sugar percentage is high, then water percentage should be low, right? If water percentage high, then sugar should be low. And that's how we can make 100. So every time it goes like that way, okay, to make 100%. So basically what I want to say, so if you increase the solute percentage, if solute goes high, that means solvent means the liquid, basically, that needs to go down. That percentage needs to go down, right? I hope you all are going to agree this one. So that's the main formula we are going to apply over here. So what happened now? Now let's say This is a bacteria. Okay. And over here we have 30% sugar. Outside over here, it's a sugar solution inside your mouth. You had lots of chocolate and all this sugar, you know, maybe Pepsi, Coke, and all those. So inside of your mouth environment over here, it's 70% sugar. Okay. So if this is the situation and bacteria are inside that environment, so this is the environment. Let me, okay. So this is the area. Outside has 70% sugar. Inside of a cell, prokaryotic cell, that has 30% sugar. So solute concentration is high over here and solute concentration is low over here. Right? So if we go by the definition of diffusion, so what it's supposed to do, that sugar must need to flow from outside to inside, right? But practically it does not happen because of the plasma membrane. Okay, cell membrane does not allow. So basically the solid particles cannot move like this way if there is a permeable membrane. If membrane is not there, like as I told, I gave you an example, like inside a room, you can spray the room freshener. So there is no divisible membrane in between. So solid particles can move. But in this case, we have a plasma membrane. It's a semi-permeable membrane. It won't allow to move solute that easily. So liquid, in this case, liquid is actually going to move from inside to outside. So if we consider this cell, okay, the bacteria, what will happen? So 30% sugar means obviously we have 70% water inside, okay? But over here, 70% solute means only 30% water outside, okay? Now water concentration is high comparing this area. So what will happen? If the situation is like this, then water will move from high concentration to low concentration. So basically water will go outside from the cell. So prokaryote, that bacteria is going to lose the cell or lose the water. Okay, slowly it will be dehydrated, it will shrink. So that's the situation over here. So what happened over here? So solute concentration is high, very high regarding inside. So solute concentration is high. That actually tells us the liquid concentration is low. And that's the reason over here we have low solute concentration and high liquid concentration. And that's the reason the water is moving from inside to outside. So basically in this case, the high solute concentration actually causes the movement of the liquid. And it will create a high pressure. You can understand that water is low, water is high over here, and that cell is going to lose all the water. Okay, that creates, this is called the osmotic pressure. That osmotic pressure occurs because of the solute concentration, differences in solute concentration between outside environment and inside body environment. Okay, so that causes the movement of the water. In this case, what will happen? The bacteria is going to lose all the water, not all, but lose a huge amount of water. It will be dehydrated. The same thing happens when you go into the sea. If you are under the water, sea water for a long period of time, you get dehydrated. Why? Because the salt concentration or the solute concentration is much higher outside in the water. Okay, so that's why your body cells, they actually going to lose all the body water. And that's why you feel dehydrated. If you do not drink enough amount, sufficient amount of water after that, then you will be very sick, because we need a huge amount of water inside our body. Okay, so over here, this situation, and this causes the osmotic pressure. Cell wall actually helps or protect all these organisms who has cell wall from this osmotic pressure. Okay, so when this is the situation, so outside environment, solute concentration is really high, so that condition is called hypertonic solution. Okay, hypertonic solution, hypertonic means solute concentration is high outside. Every time we go by the outside environment. Okay, so comparing the inside body environment, outside we have more solute. So that's why this solution is called hypertonic solution. So any cell, when they are inside a hypertonic solution, what will happen? That cell is going to lose water. Accordingly, completely opposite way, if the cell in hypotonic solution, mean outside the solute concentration low. So if it is happen like, let me draw over here. So we have let's say 30% solute, means 70% water, but over here we have 70% solute and 30 water. Remember, I told that solute, the solid particles will not move, water will move. What will happen? So outside here 70 water, over here 30. So from outside to inside, water will move. The cell will grow inside, and ultimately at certain period of time it will burst, because all cell has capacity to carry carry the water or the substances, any substances. So after a certain period of time, it will burst. So this situation happens for each and every cell. Now, if the cell wall is there, like if you get a red blood cell from our body, this thing happens. Okay, and in that case, what happened when our animal cells, we do not have cell wall. So animal cells, when they are in hypertonic solution, that cell will shrink slowly, will shrink, and this is called crenation, and ultimately it will die. Hypotonic solution, in hypotonic solution, what will happen? The cell will increase in size, and ultimately it will burst, and this is called cell lyse. Cell will burst. Okay, cell wall of bacteria actually protects not only bacteria, plant cell also, that's eukaryotic cell, but the same situation, osmotic pressure occurs in same way. So what happens? The cell with cell wall in hypertonic solution, look, the plasma membrane started shrinking, okay, but the cell wall remain intact. So it actually, so for long period of time, that cell will survive. Cell will remain alive. Okay, it's not like our animal cell. Animal cell will die very quickly, but plant cells and bacterial cells who has that cell wall, they can actually survive for a long period of time. And this process, like the plasma membrane, actually gets separated. It shrinked and it become detached from the cell wall, but the cell wall remain intact. Okay, and it protect the inside environment. So that's in hypertonic solution. Hypotonic solution, again, that cell wall, it's called lysis, same like that. It will grow, it will become, it will enlarge in size, but cell wall actually help it to be survive for long period of time. But both the cases, this is the condition I told you in hypertonic and hypotonic solution that occurs for each and every cell. And the cell wall, prokaryotic cell wall, actually, that's a protection from this osmotic pressure. And remember, I told you that humidity is very important. Okay, so most of the time they need that aquatic environment. Okay, and that actually the cell wall actually protects them in that situation. Now we are going into the details of all the cell organelles. So first, the nucleoid region, I told you that the prokaryotic cells, they do not have nucleus, but there is a specific area where we can see the chromosome, okay? Chromosome associated with some type of protein. They don't have histone protein, but they have some protein which is associated with the DNA. The chromosome, for prokaryotic cells, most of the bacteria, they has just one large, round, and double-stranded chromosome. Okay, so circular, and double-stranded chromosome, and number is 1. Always they have just one chromosome. Plasmids, they are actually chromosome. Okay, these are called extra chromosomal materials. Okay, extra chromosomal materials means they are chromosome. Actually, they are chromosome. Okay, but very short chromosome. They are also small, very small. Comparing the actual chromosome, it's really large. Let me write it down. It's large. It's very small and double-stranded chromosome. But multiple plasmids are there. Okay, the number varies from 100 to 1,000. It's multiple. Now they are actually chromosome, but they are not truly the chromosome, and that's why they are called extra chromosomal materials. Why? Because these chromosomes, they are the genetic material. They carry the hereditary information. Okay, but these plasmids, they don't carry any hereditary informations. Okay, so that's why they are extra chromosomal materials. But these plasmids are very, 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 very important for the survival of the bacteria. Why? Because these plasmids carry few protective genes like antibacterial genes. They carry. Usually inside the plasmid, as I told that multiple plasmids are there, but very small inside, double-stranded. At the max, 50 genes are there inside one plasmid. Okay, but over here, at least 10,000 genes are there. So you can understand that this is huge. This is very large, and comparing this one, this is very small, minute one. But for their existence, these plasmids are very important. They are the extra protection. Okay, like antibacterial genes, they carry these antibacterial genes. Okay, and the bacteria who has any plasmid that has antibacterial genes, you cannot kill that bacteria with that, sorry, not antibacterial, antibiotic. I'm so tired now. Antibiotic genes, okay? So basically, what happened, let's say, this is antibiotic genes. So basically, let's say, this is a small plasmid, okay, double-stranded small plasmid. Only 50 genes are there at the max. It could be 5, it could be 10, it could be just 2. So let's say this portion is, let's say the any type of antibiotic, streptomycin kind of, let's say it's streptomycin antibiotic genes are there. If bacteria with these antibiotic resistant genes, actually, it would be very difficult to kill that bacteria with that antibiotic. Okay, so that's why these are the protective organelles for the bacteria. So antibiotic resistant genes. Okay. So I just talked about only antibiotic resistant genes. There are a few other very important genes plasmid carry that's very important for the survival of the bacteria. So that's why plasmids are very important. And remember, they're extra chromosomal materials because they don't carry any hereditary information. Okay. Then the ribosome take part in protein synthesis. Okay, this is the place where cell makes the protein. For prokaryotic, the weight of the ribosome is 70S. Ribosome has 2 subunits, large subunit and the small subunit. For prokaryote, the small subunit is 30S, the large subunit is 50S. Together, don't try to add 50, 30 together gives you 80, but we don't add. Okay, it's S represent Svedberg. Is actually Svedberg unit. It's how much it's sedimentary unit actually, and completely different. So anyway, that gives us 50S plus 30S, that gives us 70S ribosome. For us, for eukaryote, it's 80S. Okay. We have 40 plus 60S. Together, we have 80S subunit. But for the prokaryotes, that's the 70S. And this is the place of protein synthesis. So what's the time? Do we have time? Yeah, we do. Okay, so now the inclusions bodies. Inclusions, these are actually storage vesicle. Okay, so inclusions, you can understand from the word, the inclusions. So extra, these are vesicles. Vesicles means membrane bound sac-like structure. But remember, this membrane is just one. Okay, they are not double membrane structure. So these are the vesicle, or you can say these are the storage vesicle. What they store? They store nutrients, extra nutrients they actually carry, or you can say important nutrients they store inside all these storage vesicles. Okay, they store glycogen, starch, all these nutrients they actually store. Different inclusions body actually store different type of materials. Like over here in this picture, B, where is B? B, that's the volutin granules, metachromatic granules. This volutin, it's also called metachromatin granules. So basically, these vesicles, they store inorganic phosphate. Okay, they store inorganic phosphate, and this material, this inorganic phosphate, is very important to make biofilm. It helps them. There are few bacteria like Corynebacterium. This bacteria, they carry this volutin or metachromatin granules, okay? And they store that inorganic phosphate so that they can make biofilm easily. Biofilm, I hope you all know that's a plaque. Like inside your teeth, you get plaque, right? So that plaque means that's the biofilm of different microbes. It's not only bacteria. There could be bacteria, protozoa, fungus, viruses, so many different type of microbes together. They actually change their identity and they convert themselves into one organism, okay? So that is the biofilm. Very, very bad for our health. That biofilm from your teeth that can go inside, if that enter inside your circulatory system, means inside your artery, it can block the artery or vein, even the heart, okay? That can block. So it's very bad. And the bad thing is it's very difficult to kill that biofilm, because you can understand that it takes long time to identify the identity of all those organisms together, okay? Multiple different microbes are there. To make that biofilm, they must have that inorganic phosphate, and this bacteria, Corynebacterium, they store that inorganic phosphate inside that inclusions body. Then what else? Oh, polyhydroxybutyrate droplet. So polyhydroxybutyrate, PHB, it's called PHB. That another like E. coli, Pseudomonas also carry. So these are the bacteria. They actually store degradable bioplastic. They store something that we use to make bioplastic we are using. So very, very important, okay? So we are collecting these bacteria to make our degradable bioplastic from all those PHB, okay, polyhydroxybutyrate droplets. So these are the inclusions body. Based on their need, they store extra nutrient material inside all those vesicles, or vesicle means it's a sac-like structure. Next one is the endospore. Endospore, remember I told that not all bacteria carry that endospore, okay? Endospore, so basically the bacteria, there are two stages, okay? One you can say the vegetative cell. Vegetative, this is V, vegetative cell. That's the actual bacteria that can grow and can reproduce. So that's the vegetative body for any cell, which can grow and which can reproduce. That's the vegetative cell, okay? So whatever under the microscope we see, that's the vegetative body or vegetative cell. Now, there are few bacteria when the environment is not favorable, okay? Like when you spray antibiotic or any disinfectant, so lots of poison outside, okay? Those bacteria, what they do immediately, they convert the vegetative body into this endospore. Okay, what they do, they multiply their genetic material. The first target is to copy the genetic material, and they outside of that genetic material they make multiple layer, and it's resistant to heat, resistant to acid, resistant to kill the cold. So it's very difficult to kill that spores. And these spores basically, it's their protective structure, okay? And they remain as endospore for millions of years. You can boil, you can apply acid, it's very difficult. They can survive any high temperature, high altitude, high acidity, so it's very difficult, and they can stay like this for millions of years, okay? So this is actually when one vegetative cell converts into endospore, that process is called sporulation. Okay, so from vegetative cell, they convert into spore, and that process is called sporulation. Now, this endospore, they does not have capacity to grow. They does not have capacity to reproduce. Okay, so that's the difference between a vegetative cell and one endospore. Now, over here, there are steps. What are the different steps they do to convert themselves into one endospores? So first thing, they replicate DNA. That has to be there because that's the main target, right? To protect the genetic material. So they multiply, they make the copy of their DNA. They just have one DNA, so it does not take that much time. Then outside of the DNA, they make septum. Okay, so this is called septum. So just one membrane. So when that septum surrounds the genetic material, then this structure is called pore spore. So basically at this moment inside the vegetative body, we have a pore spore and we have our genetic material. This is the nucleoid region, okay? Then what they do outside of this pore spore, they have to separate the pore spore from the mother cell. This is the mother cell, right? So then they make one more membrane, additional membrane they make, okay, over here, and that separate the pore spore from the mother cell. Now, immediately after the second membrane, so septum is the first membrane, okay? So septum, that's the first membrane outside of the chromosome. Then second membrane they make, additional membrane or second membrane, okay, that's the second one. Then immediately outside of that second membrane, they make cortex, another membrane. These membranes, they actually outside of this second membrane, they deposit calcium, cholic acid, it's called dipocholinic acid. So all these things, they add up, basically to protect. Okay, so it's very difficult to degenerate this calcium and dipicholinic acid and all those. So these are the protection. It's very hard to destroy, to break, to dissolve this layer, the cortex layer. So that's the third layer. Then outside of the cortex, they provide protein layer. Protein layer means that's the supply of food for the chromosome. Okay, inside, how are they going to be survived for years after years? Okay, so that's the reason they make a protein layer to provide the food for the DNA. Okay, for the endospore. Then when the protein layer or protein coat is formed, then finally outside of the protein layer, so everything is there, the protection, you know, so many membranes they make to protect the DNA, you can understand. Then finally, they make, it's called exosporium, okay, outside of the protein layer. Once that exosporium is formed, then that endospore, then it's endospore. Okay, we started with pore spore. Now, after formation of exosporium, that pore spore convert into endospore. Okay, now that endospore, the cell actually lies, the original mother cell, it dies and it releases the endospore. Okay, so this thing happens when the environment is not favorable. When you spray disinfectant, when you spray Lysol and all those, so they just, what they do, original bacteria dies, but they release the endospore. Okay, now when that environment is favorable, after certain period of time, this endospore, they can convert into vegetative cell. Okay, so basically they convert themselves from the spore, or the bacteria, the mother cell can be originated, and this process is called germination. Okay, so by the process of germination, when the environment is fine, nice, enough food supply is there, oxygen is there, humidity is there, nitrogen supply is there, then all these endospores, they germinate and they convert themselves into a vegetative cell. Means now they can grow and they can reproduce. So that's the endospore. Now the plasma membrane. Okay, so basically this is the first layer. Okay, immediately outside of the cytoplasm, prokaryotic cell has the plasma membrane. Plasma membrane, as I told, that all the chemical activity, all the metabolic activity, not the chemical, all the metabolic activity is going to occur over here. The plasma membrane or the cell membrane is made up with phospholipid bilayer and the protein. Now the phospholipid, it's a phospholipid, it looks like it has a head and a tail. Okay, and this is called phospholipid. Phospholipid means it's a lipid plus phosphorus. The head is made up with phosphorus. We are going to study that. So this tail is made up with lipid and the head is made up with phosphorus. So membrane, plasma membrane has phospholipid bilayer, means 2 layers of phospholipid. So basically like this. So the arrangement is, every time the arrangement is like this. So you can see that all the tail part, you can notice over here, the tails are together, heads are separated from each other, because this head part, it can tolerate water, but the tail, this part cannot tolerate water. Okay, so that's the reason all the tail part remains inside. The area that cannot tolerate, that does not go with the water, that remains inside. So the outside environment is watery and inside environment watery, that does not affect the bacterial body. Get it? So that's the reason that head remains both inside and outside, and that's why the phospholipid arrangement is like this. So and 2 layers of phospholipid, so that's why it's called phospholipid bilayer. Mostly the phospholipid bilayer, and inside the phospholipid bilayer we have so many different types of proteins. The function of plasma membrane, this is the semi-permeable membrane. It controls the movement of particles, okay, who can go in or who can go out. It is controlled by the plasma membrane. If the plasma membrane is not there, if you take out the plasma membrane from any cell, that cell is going to die, because all the solute, you can understand easily that osmotic, the concentration, osmotic pressure will be different. The cell may not tolerate salt. The salt from outside environment come on in. All the organic, inorganic ion concentration. So it would be bizarre situation actually if plasma membrane is not there. So inside the phospholipid layer, as I told that there are so many different types of proteins. So protein, the function of protein, what they do, they maintain the relationship with other cells. That's number one, means communication with the cell. They do that, and these proteins are responsible. Any type of information that needs to go from outside to inside, these proteins, they do. The transportation, all the food materials, any materials that these proteins, they are responsible to transport from inside to outside, outside to inside. Okay. They are actually glycoprotein, they are responsible for the identity. Okay, by seeing, you can see these are the glycoprotein. This is the ID tag. Each and every cell has specific type of glycoprotein that identify that special person or special organism. Like I have my own glycoprotein outside of my cell, and nobody else has that glycoprotein. Okay, that special type of protein, that's my ID tag. Okay, by identifying that protein, you can identify me. Like my it will not match with anybody else, so that's why it's called ID tag. So that glycoprotein, that's the ID tag. So all the functions and all these cells, protein are actually the site of all the chemical reactions. Okay, so these are the functions of protein. So basically they communicate with other cells. They are the anchoring organ. They are the attachment side of each cell. They hold the other cells together. This is the site of chemical reaction, that number 3. Number 4, they are the ID tag. Number 5, they control the movement of the material substances. They transport food inside, or they transport that cell does not need to outside. So all these are the functions of protein. So most of the cellular function is actually done by this protein, not the phospholipid bilayer. Okay, phospholipid bilayer has a different function, but they actually carry that protein. So the structure of plasma membrane, it's called fluid mosaic model. If you see under the microscope in plasma membrane, it looks like that it's a fluid, semi-solid structure. All these protein components, protein, glycoprotein, whatever the organelles, not organelles, the compounds are there inside the plasma membrane, they can float inside that phospholipid bilayer, because of the presence of cholesterol. Okay, this inside this phospholipid bilayer, I don't see over here, but we have cholesterol over here inside the phospholipid. One of the reasons of this fluidity, so that the proteins can float, is the cholesterol, because of the presence of cholesterol. That one reason. Another reason is that phospholipid bilayer. What happened? Phospholipid, it has 2 fatty acid chain. These are the fatty acid chain. Okay, these 2. Now, inside the phospholipid, few of the fatty acid chains they have kink, like they are not straight, okay, or not tightly packed. They have bend inside, so they are not tightly packed. Okay. We will study about the phospholipid structure in later chapter, so I'm not going into the detail, but just to let you know that if there is a kink over here or bend, so all these fatty acid, they cannot arrange themselves tightly, okay? In between the particles, some gaps will be there, and that makes the structure a little bit fluid. That gives the fluidity. So these are the two reasons, okay? So unsaturated fatty acids and the presence of cholesterol, okay? You can say over here, this is called unsaturated fatty acid, okay? So this is the reason 1, and this is the reason 2, because of this fluid mosaic model. So that's the reason behind this fluidity of the plasma membrane. Now the mosaic, if you see that plasma membrane from the microscope, it gives a nice pattern, you know, like inside that phospholipid, that these different shapes and types of proteins, they are floating, so it gives a nice pattern, and that's why it's called mosaic. It looks like mosaic pattern. So that's why this model, or the structure of the plasma membrane, it's called fluid mosaic model, or fluid mosaic pattern. So that's the plasma membrane. I already talked about it, the diffusion. This is the simple diffusion. So particles move from the high concentration, this is the high, to low, okay? So that's the simple diffusion. Facilitated diffusion is, it's a diffusion. In this case also, the materials or the particles move from high to low, but what happened in this case, the materials cannot directly come through the membrane, okay? In this case, one protein, remember I told this, all there are few protein substances who actually help to transport materials. So these protein who actually help in transportation, they are called carrier or channel protein. So carrier protein, or they are also called channel protein. So channel protein usually carry the water, and carrier protein everything, including iron. So when these carrier protein, they move the particles from high concentration to low, that is called facilitated diffusion. So basically, same thing happens like simple diffusion, but this carrier protein, they are helping the movement, okay? So that's why it is called facilitated diffusion. In case of active transport, what happened? This is completely different than the diffusion. In this case, what happened? The particles move from low concentration to high concentration. Completely opposite of diffusion, okay? Now to do this, so you have only let's say 3 candies you have, and you have to distribute that hard candies to 10 people. So you can understand what you have to do. In this case, you have to spend money, right? So you have to buy more, or you have to do something. So here, to do that, when you have a less amount than you need, then cell does this active transport. In this case, all this carrier or channel protein, they transport material from outside to inside, okay? And in this case, all this thing happens with the help of the ATP. So that's the energy, okay? It's like money. So over here, you have to buy another 7 candies, right? So for that, you need money. So exactly same way, to do that, cell needs ATP, okay? So for diffusion, we really do not need any energy. So no need of ATP. We do not need energy for the diffusion, but over here for the active transport, we must have supply of ATP. And in this case, the particles move from low concentration to high concentration. So that's the diffusion between active transport and the diffusion. Cell wall, what's the time? Okay, I have,
5:54.
yeah, so we have another 4 or 5 minutes. Let me finish this part, the cell wall. So cell wall of prokaryotic cell, very, very important, okay? It is made up, the main component of cell wall, prokaryotic cell wall is called the peptidoglycan. So you can see this is the cytoplasm. Then outside of the cytoplasm, we have plasma membrane. Then outside of the plasma membrane, we have that peptidoglycan for the gram-positive bacteria. So this peptidoglycan, it's a huge polysaccharide, large carbohydrate. It's a long chain, large carbohydrate, and it looks like long chain made up with alternating NAM and NAG molecule. NAG is N-acetyl glucosamine, that's another sugar, and NAM, that N-acetyl muramic acid, that's another sugar. This alternating, you can see that's blue at the NAG and yellow at the NAM. So alternating NAG and NAM molecules, they together make a long chain. Okay, so multiple long chains are made up with alternating NAG and NAM molecules, and all these chains are connected with bonds, different types of bond. In case of this is the gram-positive bacteria, we have you can see the tetrapeptide. This is 1, 2, 3, 4 peptide bonds associated with 5 pentapeptide bonds. Okay, so basically 9 peptide links are there. So tetrapeptide is connected with pentapeptide, and together penta and tetra connect all these long chain of NAG and NAM. So basically they make a mesh-like structure. How they arrange like long chain, okay, and then underneath the chain we have this pentapeptide, tetrapeptide. So basically it's a mesh-like compound, thick structure. For gram-negative bacteria, everything is same, but all these chains are directly connected with the tetrapeptide. So that pentapeptide is absent over here. Look, in this case, you can see only 4. So these chains are only connected with tetrapeptide bond. So that's the peptidoglycan. Okay, so this is the structure of peptidoglycan. Now, based on the thickness, how thick the peptidoglycan layer, based on, we have gram-positive bacteria and gram-negative bacteria. Very, very important. For gram-positive bacteria, this peptidoglycan layer is 30 to 100 nanometer thick. In case of gram-negative bacteria, this peptidoglycan layer is only 2 to 4 nanometer. So basically, what's the difference between gram-positive and gram-negative bacterial cell wall? In case of gram-positive bacteria, the peptidoglycan layer is very thick. But in case of gram-negative bacteria, they have a very thin, very little amount of peptidoglycan layer. Okay, but both the cases, the cell wall is mainly made up with peptidoglycan layer. Now there are little bit more differences. What happens? So if this is the gram-positive bacteria, you can see immediately outside of the cytoplasm we have plasma membrane, right? Cell membrane. Immediately outside of the cell membrane, we have a thick peptidoglycan layer. And outside of the peptidoglycan layer, towards the end of the peptidoglycan layer, gram-positive bacteria, they carry teichoic acid or teichoic acid. Okay, this teichoic acid actually is responsible for the pathogenicity of the bacteria, and basically the virulence and all those things are dependent on this teichoic acid for gram-positive bacteria. So gram-positive cell wall is very simple. Outside of the plasma membrane, they have a thick peptidoglycan layer associated with the teichoic acid. Now the gram-negative bacteria, they have, this is the cytoplasm, this is the plasma membrane. Immediately outside of the plasma membrane, they don't have peptidoglycan layer. They have a over here, they have a gel-like space, and that's called periplasmic space. And outside of the periplasmic space, they have that very thin peptidoglycan layer. So for gram-negative bacteria, they have periplasmic space that is absent in the gram-positive bacterial cell wall. Okay, we don't have any periplasmic space over here. So basically, plasma membrane and peptidoglycan, they are almost attached. Over here, we have jelly-like substances that makes the periplasmic space. Then after periplasmic space, very narrow, thin peptidoglycan layer. But we have something else outside. That's not the only layer, the peptidoglycan layer, just like, gram-positive. Outside of the peptidoglycan layer, they have another layer of cell membrane. So in this case, two layer of cell membranes. So this cell membrane or this layer of plasma membrane is called inner membrane, and this layer of plasma membrane is called outer membrane. Okay. Now the outer membrane is associated with lipopolysaccharide, a large carbohydrate complex. Okay. So gram-positive bacteria, they have teichoic acid. Gram-negative bacteria, they have lipopolysaccharide, which is responsible for pathogenicity and the virulence. Okay, very, very important. So gram-negative bacteria, you can understand that their cell wall is really complex. Okay, and they have so many protective structure. They outside of the peptidoglycan is covered with the outer membrane. Then they have lipopolysaccharide. Now lipopolysaccharide has three parts. So this is the O antigen, this is the fatty acid chain, and this is the main carbohydrate. Okay, so these are the three parts. So O antigen, this part means the outer portion. So that this fatty acid, it's called lipid A. This lipid A remains attached with the outer membrane. Okay, so this part, this is the root. Okay, so lipid A part remain attached with the outer membrane, and O antigen, they are responsible for all this pathogenicity of virulence like septic shock syndrome and all those that comes from the gram-negative bacteria because of the presence of this O antigen inside that lipopolysaccharide. Okay, so these are the differences between gram-positive bacteria and gram-negative bacteria. So first difference, the thickness of the peptidoglycan layer. Then number two, gram-negative bacteria immediately outside of the plasma membrane, they have periplasmic space, and this layer of plasma membrane is called inner membrane. They have periplasmic space. Periplasmic space is absent over here. Peptidoglycan is very thin. Then they have outer membrane. Outer membrane is absent over here. And the gram-negative bacteria, they have lipopolysaccharide, but gram-positive bacteria, they have teichoic acid. Okay, so that's the cell wall. So one group of bacteria, it's called acid-fast bacteria. They are actually gram-positive bacteria. You can see the peptidoglycan layer is very thick, but along with the teichoic acid, they have mycolic acid. It's a soap kind of structure, okay, wax kind of structure, you can say. They are completely different types of organism, and they are called acid-fast organism. They don't take gram stain. Okay, for them we use acid-fast staining mechanism, and they are called acid-fast bacteria. So all the acid-fast bacteria, they carry the gram-positive bacterial cell wall structure, but they carry mycolic acid. Okay. So we are done. Okay, so up to this today, and I will complete this chapter Wednesday. Okay, so do you have any question? No? Okay. Okay, so I cannot. Okay, so good night then. Let me end it up. Okay. Good night. If you have any question, you can ask me. I'm still here. James, do you have any question?
Oh, sorry, no, good night.
No? Okay, good night.